I'm delighted to see everyone here this afternoon. I'm Rick Lifton, the president of Rockefeller University, and it's a wonderful pleasure to welcome you all to the 66th Convocation of the Rockefeller University. It's great to have so many of you here with us in Casper Auditorium for this auspicious occasion. So if this is your first visit to Rockefeller, you may have become aware, because we're not shy about saying it, that we're a different kind of university. We're completely focused on a singular mission, science for the benefit of humanity. This dates back to our founding by John D. Rockefeller in 1901, who recognized that the biggest obstacle to preventing or treating diseases was the lack of understanding of their fundamental causes. By design, we're a small institution with only 70 heads of laboratory, but these 70 are selected for remarkable promise to do extraordinary science that will transform their fields. And they're given an abundance of time and concentrated resources and a wonderful environment in which to achieve their audacious goals. We do not have undergraduate students. Our students are all pursuing Ph.D. degrees in biomedical sciences, and were accepted based on their demonstrated intellect, scholarship, creativity, and drive that make them among the 4% of all applicants that we accept into our program each year. This clarity and focus is a tremendous source of our strength. It inspires an amazingly talented faculty to pursue the most audacious goals in order to have the greatest impact. We also have a board of trustees whose passion for innovation matches that of our scientists, and pertinent to today's festivities, a supremely creative and dedicated group of doctoral students. The results of our high aspirations and expectations speak for themselves. There have only been 258 professors in our history, and 26 of them have won Nobel Prizes, and another 25 have won Lasker Awards. These include 6 Nobel laureates in medicine or chemistry in just the last 25 years. These totals are not matched by any other institution, regardless of size. Discoveries by Rockefeller scientists include the causes and novel treatments of many diseases, as well as fundamental insights into basic biology. These include one of the key discoveries in the history of biology, namely that DNA is the chemical of heredity. Differences in DNA sequence are recognized to determine whether a life form is a human or a mouse or a plant or a bacterium, and variation within species determines why we don't all look like one another and why some of us are more susceptible to one disease or another. And this knowledge over the last 80 years has been put to great use for the prevention of disease, ranging from the vaccines that prevented millions of deaths from COVID to the cure for hepatitis C, for which our colleague Charlie Rice received the Nobel Prize, to environmentally cleaner manufacturing processes, as pioneered by one of this year's honorary degree recipients, Francis Arnold, and led to the discovery by a graduate of our Ph.D. program, Svetlana Moisov, of the peptide hormone GLP-1, which has recently become renowned for its remarkable ability to produce weight loss and markedly reduce heart attacks, the development of kidney failure, and prevent death in people with obesity and diabetes. Since the first 5 Rockefeller PhDs were conferred in 1959, graduates of our PhD program have gone on to highly impactful careers at the world's leading universities, in the biotechnology and pharmaceutical industries, and in other areas of commerce in which strong analytic skills are of great value. Today, we are immensely proud and excited to be conferring the PhD degree upon 38 graduating students. These truly exceptional individuals have been integral members of our community of scholars who, through their inspiration and hard work, have become outstanding scientists in their own right. We recognize the amazing talent and resolve these graduates showed during the conduct of their doctoral research, during which, you may recall, they persevered through the extraordinary disruption of the COVID pandemic in the middle of their time here. Our convocation is also the occasion on which we recognize individuals who have made exemplary contributions to the advancement of science and the betterment of humanity. We do this to honor these individuals for their remarkable achievements, but as importantly, we do this to present these distinguished individuals to all members of our community and to the world as exemplars of the impact one can have through a life dedicated to science, the pursuit of truth, and service to humanity, and to set a standard to which we can all aspire. This afternoon, we will confer the degree of Doctor of Science honoris causa upon 3 individuals who are indeed extraordinary exemplars of the Rockefeller Credo and who are inspirations to all of us. Drs. Francis H. Arnold, Dr. Freeman A. Hrabowski III, and Dr. Michael Mann. We are here today due to generations of leadership by the Rockefeller family, with our graduate program being uniquely attributable to the dedication of David Rockefeller, who recognized the importance of ensuring the continuation of science by educating new generations of scientific leaders. And he understood that these young scientists also bring energy and new ideas to our own laboratories. David was committed to the graduate program's success and deeply involved in every aspect of its development, and our graduate program proudly bears his name. Because of the university's small size, we thrive without departments and disciplinary boundaries, and this allows our students to be immersed in a highly interactive environment. They learn from incredible scientists working in diverse areas using distinct technologies and approaches, and they also learn a great deal from one another. Nonetheless, their closest collaborator is their mentor, and we're very proud of the keen commitment our heads of laboratory have to the success of their students. This commitment stands at the heart of the graduate program. Today's ceremony highlights this relationship as the achievements of our graduates are celebrated by their faculty mentors. Our graduates today, like their predecessors, have learned science by doing science. They've made many important discoveries in their time here, and I hope all have shared the awe of discovering something new about fundamental life processes, or have had an insight that will advance the diagnosis or treatment of a disease. As you'll hear this afternoon, they've been remarkably creative and productive, and we are immensely proud to graduate them into the Academy of Rockefeller doctorates. Our graduates, of course, now will go on to the next stages of their careers. The pace of technological advances in biomedicine over the last 50 years have been nothing short of astounding, leading to development of countless opportunities to gain fundamental new insights into biomedicine that will benefit humanity. I trust that all of our graduates will carry with them strong powers of observation, technical prowess, and analysis, and a deep belief in the importance of dispassionately seeking the truth and distinguishing what is true from what could be true and what is clearly false. Amid a rising time of poorly thought out arguments that constitute misinformation in our broad public discussion and intentional fabrications solely meant to mislead the public, that is disinformation, our graduates' skills and the integrity to use them for public benefit will make these individuals important keepers of a flame that is critical to sustaining a civil and productive society. We are immensely proud of all of our graduates' accomplishments and look forward with high expectations and optimism to your next chapters. Congratulations to this year's graduates.
Good afternoon and welcome. Mr. President, Mr. Chairman, graduates, Rockefeller community, and honored guests. This ceremony is part of our unique tradition at Rockefeller of focus on our graduating students. I now give my formal assurance to the President and to the Board that these graduates have satisfied their obligations and are eligible to be conferred the degree of Doctor of Philosophy.
Good afternoon, everybody, and I want to welcome once again everybody to Rockefeller and thank you for being here to celebrate our graduates. Dalton Ban. Dalton graduated from the University of Southern California and joined the tri-institutional MD Ph.D. program in 2018. The same way we get sick from viral infections, bacteria are also infected with viruses and need to develop immunity against them. In my lab, we investigate how bacteria defend from their viruses using an immune system called CRISPR. Dalton has a prominent scientific curiosity. Dalton's first project studied how CRISPR works in the human pathogen Staphylococcus aureus. But this curiosity led him to look beyond CRISPR and focus on another antiviral system of staphylococci, in particular the CVAS systems, which uses a CGAS cyclase to start defense. Dalton first trained and then worked along with another student in the lab, Cameron Roberts, to investigate how bacterial viruses activate the CVAS immune response, something that has been a mystery since the discovery of this immune system many years ago. Dalton and Cam made the seminal discovery that phages produce a small RNA molecule that activates defense. This is the first time that we study something that's not CRISPR in my lab, and therefore Dalton will be credited with opening a new and exciting area of research in my group. Mr. President, Mr. Chairman, and honored guests, it is my pleasure to present to you Dalton Mann.
Congratulations.
It is my pleasure to present Adi Berman on behalf of Professor Tarun Kapoor. Tarun provided the following comments. In our lifetimes, 2 copies of our DNA are divided between daughter cells over 10,000 trillion times. During each cell division, micrometer-sized protein polymers called microtubules provide the mechanical framework to physically separate the DNA in the form of chromosomes and ensure that 1 complete copy of the genome is transferred to each daughter cell. Errors in this process, which takes only minutes, can lead to miscarriages, developmental defects, and cancer. Adi creatively combined protein biochemistry, electron microscopy, and live cell microscopy methods to examine how microtubules are formed in human cells. She characterized a multi-protein complex that functions as a template for microtubule assembly. Using a series of innovative experiments, she uncovered how each microtubule's growth and shrinkage is controlled in space and time, and how each polymer is anchored within the cell to provide the forces needed to transport chromosomes. Her work uncovered a fundamental mechanism that ensures the stable propagation of our genomes through cell division and reveals how mutations in the proteins that control where and when microtubules form may cause disease. After completing her thesis research, Adi obtained a highly sought-after postdoctoral position at Regeneron Pharmaceuticals. During her time with us, Adi inspired us with her ingenuity, tenacity, and intellect. She has an impressive ability to reason through complex data, develop testable hypotheses, and design experiments to illuminate essential cellular mechanisms. We will miss Adi's excitement for cell biology, for dynamic protein polymers, and her skill at balloon sculpting. We thank Adi for helping to establish a tradition in our lab to celebrate each graduation with a balloon sculpture that commemorates the main scientific achievement. The art, along with the unexpected popping of the balloons, continues to provide joy and lasting memories. We wish her the very best and look forward to her continued success. Mr. President, Mr. Chairman, honored guests, it is my pleasure to present to you Adi Berman.
Congratulations.
Sarah Kai, trained in structural biology at Caltech. Sarah joined the lab exactly 5 years ago at a time when Hiro Takai in my lab had just realized that telomerase, long held to provide the solution to the so-called telomere end replication problem, is actually not sufficient to keep our telomeres intact. Hiro discovered a second end replication problem that is not solved by telomerase, but is solved by an enzyme called CST pol alpha primase. You'll hear more about that. Sarah focused on this second telomere maintenance enzyme, aiming to understand how it works and how it gets to the telomere. She chose to use cryo-EM to determine the structure of CST Pol-alpha primase, but immediately faced a hurdle. Her thesis advisor, me, knew nothing about this technique and obviously could not advise her. And like many other hurdles she would later face, Sarah rapidly solved her problem by asking our neighbor and cryo-EM expert Tom Waltz to become her co-advisor. What followed was a breathtaking period in which Tom and I witnessed plenty of downs, but many more ups in Sarah's path to her Ph.D. She showed not only what the enzyme looks like, but eventually solved the puzzle of how it gets to telomeres. Her work taught us that this enzyme is brought to telomeres by the POT1 subunit of Sheltrin, and her structure explained how inherited mutations in CST or POT1 lead to devastating telomere disease. But wait, there is more. She also showed that phosphorylation of POT1 regulates the enzyme so that it can do its job at the right place in the right time. Sarah is a superb experimentalist, a creative thinker, a lucid speaker, and a clear and fast writer, so that Tom and I sometimes wondered whether we actually deserved to be called her thesis advisors. She combined her work in the lab with many other tasks, organizing our lab outings, working with RISI, serving on the Maine University Pandemic Committee, a huge task, and organizing several Anderson Cancer Center symposium. Her success allowed her to get the coveted Schmidt Science Fellowship, which will provide with rather generous support for her, her partner Ian, and her 2 cats during her postdoc, for which she is currently deciding between Harvard, MIT, and UCSF. Mr. President, Mr. Chairman, honored guests, it's my pleasure to present to you the remarkable Sarah Kai.
Like people, some biomolecules can seemingly do anything. SMC56 is one such example. Belonging to the structural maintenance of chromosomes, superfamily of protein complexes, SMC56 promotes genome replication and damage repair, suppresses viral infection, controls reproduction in plants, and even chemically modifies other proteins. Because of such diverse and perplexing functions, decades after it was first identified, SMC56 still doesn't have a catcher name like its closely related cousins like cohesin and condensin. The field was waiting for new light to be shed on this multi-talented molecular machine. The game was changed by a similarly multi-talented individual whom I will now proudly introduce. Jeremy Zihuai Chang was born to a family of healthcare professionals. Youngest of 4 siblings, Jeremy was so mischievous as a kid that his parents were convinced that he was a pirate in a previous life. Jeremy nonetheless excelled academically and went to college at the University of Chicago, sweeping almost all the undergraduate honors before joining the MD PhD program at the tri-institution in 2018, and to my good fortune, joining my laboratory to conduct a PhD thesis research in 2020. In close collaboration with Xiaolan Zhao's lab at the Sloan Kettering Institute and funded by a F30 fellowship from the National Cancer Institute, Jeremy took on the challenging task of dissecting the dynamic behavior of SMC56 on DNA and chromatin at the single molecule level. Through elegant experimentation and painstaking analysis, Jeremy made the groundbreaking discovery that SMC56 prefers to bind DNA at the junction between single-stranded and double-stranded regions. This property makes it unique among the SMC family members. And this discovery and a series of ensuing findings that Jeremy made provide a new and exciting paradigm to understand SMC56's many roles in genome maintenance and human disease. Jeremy's excellence goes way beyond the lab. Entrusted by his fellow trainees, Jeremy was elected the sole student representative on Weill Cornell Medicine's governing entity, the Board of Fellows. At the height of the pandemic, Jeremy volunteered to visit underserved neighborhoods and give vaccines to the most vulnerable community. Behind all these accomplishments is Jeremy's unwavering sense of responsibility, a quality that will serve him very well in the future, no matter whether he's leading an independent research lab or treating patients in the clinic. Perhaps by then, SMC 56 will finally receive a functional name, which I'm sure will have a lot to do with Jeremy's pioneering work in the past 4 years. Mr. President, Mr. Chairman, and honored guests, it is my pleasure to present to you Jeremy Chang.
Congratulations.
Ali Sahan hails from Turkey and graduated from its top university, Bilkent University, with high honors and a BS in molecular biology and genetics. In my lab, Ali worked on a leukemic fusion protein that results from a chromosomal translocation and in association with a chromosomal histone modifying complex, DOT1, acts as a rogue DNA binding transcription factor to elicit gene expression changes that result in cancerous cells. Through meticulous biochemical analyses of this complex on chromatin templates, Ali provided important new insights into mechanisms underlying the highly elevated enzymatic activity of the leukemic fusion protein DOT1 complex that was generally thought to be involved exclusively in transcription elongation. However, with insight and rigorous biochemical analyses, Ali also discovered a second non-enzymatic function of the DOT1 complex in recruiting the key transcription initiation factor, TF2D, to the template, thus indicating a novel and unexpected function of the complex in transcription initiation and opening a new frontier for the regulation by the leukemic fusion proteins. On a more personal note, Ali was not only a dedicated young scientist with an insatiable curiosity, but also a very interactive, congenial, and helpful colleague. He was an eager volunteer for organizing lab events, my constant contact for help with computer issues, and always had timely bits of interesting information. I note an instance in which I was preparing a talk for a symposium honoring my postdoctoral mentor, and Ali surprised me with a slide tracing my mentor's scientific lineage back to Galileo, and also Ali's. It's been a pleasure to witness Ali's scientific development, and he will be sorely missed, but still in the neighborhood as a postdoctoral fellow at Memorial Sloan Kettering. Mr. President, Mr. Chairman, and honored guests, I am pleased to present Ali Sahan.
I'd like to introduce Matthew Davenport. Matthew was one of those who really wanted to be a scientist since he was a little kid. Growing up in Cincinnati, Ohio, he was graduated summa cum laude at both his high school and University of Cincinnati, majoring in biomedical engineering, chemistry, and mathematics, which tells you about his interdisciplinary approach to science. At Cincinnati, he studied the neurobiology of Fragile X syndrome, a disease that causes intellectual disabilities, and published first author and multiple co-author papers for that work. He joined us in 2017 here at Rockefeller and started a collaborative project between my lab and Ali Pasha Viziri. He joined a project that was a mystery for us. We had previously discovered that the brain pathways that control the ability of what I'm doing now, speaking, in us humans and singing and learning how to speak actually in parrots and songbirds, is embedded in a brain pathway that controls learning how to move. And Matthew was fascinated by this finding and wanted to investigate the mechanism of the evolution of that relationship and whether if this surrounding motor pathway was really similar or not to the pathways that influence how we speak. So he proceeded to do a series of tour de force experiments in single-cell transcriptomics, which we call gene expression, the physiology, the electrical currents in the neurons, as well as the anatomy and the behavior and computational biology. Yes, he did all that to address these questions. And he found that the cell types within what we call the vocal learning system of songbirds do have molecular profiles that match the cell types in the surrounding motor pathways. However, they're specialized. And he found that there was one particular specialized cell type that goes from the cortex to the neurons that actually controls the voice, was the most specialized with over 1,000 genes that differed from the surrounding motor area, indicating that it is even evolving to be a new novel cell type. He also showed differences with other parts of the brain circuit that are specialized for their different functions. In his pandemic project, he took some other data and showed that this circuit that's suppressed in female songbirds, some of you have heard that only males sing in certain temperate zones and females don't. Well, females hatch with this ability, and then they lose these brain regions. And Matt, using comparative genomics, honed on in some molecules that suppress that circuit from forming in females and setting up a window for how these song nuclei develop in the first place. So from his graduate work, he has published 2 papers and another 2 that we're ready to submit. And for his accomplishments, Matthew has won a number of awards, the most prestigious ones being a Barry Goldwater Scholarship when he was an undergraduate student, and a National Science Foundation's Graduate Research Fellowship here. So Matt will be staying on as a postdoc to go dig deeper into the relationships of vocal learning pathways and the specializations of their circuits. I do want to thank his thesis committee, Corey Bargman, Priya Rajasethuputhi, and Constance Scharf for serving on this committee. And Mr. President, Chairman, and honored guests, thank you, and I'd like to introduce Matt Davenport.
Congratulations.
Thanks, Rick.
Zach Gershon. Zach grew up in Long Island, earned his bachelor's degree at Hunter College, and received just about every accolade possible by the time he joined us as a Ph.D. student at Rockefeller University, including being a Siemens competition finalist, a recipient of the HHMI and Goldwater scholarships. But it wasn't until I met him that I knew I wanted to work with him. I think it was the summer of 2017. Zach showed up in my office and he spent a good 2 hours talking about stress, anxiety, and attention, and how they're all related. He drew complicated diagrams, spoke a mile a minute, and in the end, I think his message was, attention may be a higher order cognitive function that only some animals have, but it likely came out of ancient neural circuits linked to survival, such as stress and anxiety. A little bit of stress can make you more attentive, but a lot can probably tank it. Thus, he began to carve out the beginnings of a thesis project, and right away, I loved his enthusiasm, energy, and earnestness to find a truth. For his thesis project, Zach asked the question, what shapes our individual variability in behavior? For instance, what makes some humans, or some mice, more attentive while others are less attentive? Are there single genes that have large contributions to this variation in attention? And can that point us to key cell types and brain mechanisms that enable attention in our brain, and to possibly even new targets for therapy? These were Herculean goals, but Zach was really driven to succeed, in part because of his own personal struggles and triumphs with ADHD. Over the years, Zach systematically developed expertise spanning genetics, cell biology, animal behavior, brain imaging, and systems neuroscience, I think a testament to his scientific rigor and drive. He also maneuvered many failures with patience and grace until finally landing on what I think are a remarkable series of findings. He found that the activity of a single gene and how it functions early in development dictates in large part whether mice will go on to have high or low attention as adults. How does this gene function? He went on to show that rather than stimulating brain activity, as do most ADHD medications, such as amphetamines and other stimulants, it has a striking role in quieting the brain. Thus, how quiet our brains are, even at rest, turns out to be one of the most important sources of variation that predicts attentional performance. I think a remarkable insight. Overall, Zach's work suggests that focusing less on signal amplification and more on noise reduction may be a therapeutically promising strategy to enhance attention. Notably, he accomplished all of this while leading campus community events, leading national support groups for ADHD, and in the backdrop of a pandemic and multiple international crises, each of which I know hit very close to home for him. I'm really proud of Zach's personal and scientific journey and his Ph.D., especially proud of his remarkable scientific findings. We'll miss him, his warmth, his curiosity, and his scientific drive. Mr. President, Mr. Chairman, honored guests, it's my pleasure to present to you Zach Gershon.
Olivia Goldman began her PhD wondering why insect repellents are so repellent. It turns out that DEET, the active ingredient in Deep Woods Off, is extremely bitter, leading Olivia to wonder if bitterness alone could explain why DEET is so effective in preventing mosquito bites. So she slathered her arms with the bitterest substances known to humans, but mosquitoes continue to bite her, suggesting that DEET has some other mode of action. Olivia developed methods to peer into the gene expression of single cells in search of the receptors that mosquitoes use to detect DEET. This led to the surprise that almost every mosquito sensory neuron, be it for smelling smells, tasting tastes, turns out to be polymodal. So rather than selectively responding to one kind of taste or smell, many mosquito sensory neurons seem to have the capacity to respond to temperature, noxious chemicals, odors, and tastes all at the same time. A design principle in which mosquito sensory neurons can detect everything, everywhere, all at once is likely to be very bad news for humans and our attempts to deter mosquitoes. In her last act at Rockefeller, Olivia co-led the Mosquito Cell Atlas Project, a large international single-cell RNA sequencing consortium that seeks to profile gene expression in every tissue in the male and every tissue in the female mosquito. This enormous data set is already having a major impact on the entire mosquito field. If you are attempting to fight a very bad insect, knowing as much as possible about it is very helpful. So beyond her outstanding science, Olivia has made tremendous contributions to promoting diversity and inclusion in graduate education by co-founding Científico Latino. This national nonprofit organization has successfully guided hundreds of scientists from minoritized backgrounds through the hidden curriculum of graduate admissions. Olivia was recently selected as a 2024 Schmidt Science Fellow in recognition of her science and her science leadership, and is currently conducting postdoctoral work in the laboratories of Diana Bautista and Ellen Lumpkin at the University of California, Berkeley. So although Olivia is no longer being bitten by mosquitoes, her fascination with pain apparently followed her to California because she's investigating the important and understudied problem of headache and migraines. Mr. President, Mr. Chairman, honored guests, it is my great pleasure to present to you Olivia Goldman.
Congratulations.
Amer Hossain. Amer was born in Bangladesh and moved to the US to attend Vassar College. He graduated in 2014, and in 2016, he joined our PhD program and started working in my lab. Amer worked on 2 projects. As I mentioned before, my lab studies how CRISPR systems protect bacteria from their viruses. They do so using Cas9, an enzyme that cleaves the viral DNA when it enters and infects the bacterial cell. Amer found that viruses carry DNA repair systems that fix the cleaved DNA to counteract the CRISPR immune response. In his second project, Amer made the bold move to look for novel antiviral systems in soil bacteria. He discovered a DNA glycosylase that also attacks the viral DNA by using a mechanism that is completely different than that of Cas9. Amer has been a pioneer for this last project, and the methods that he developed are now being used by half of my group. And I believe that Amer's scientific legacy will impact my lab for many years to come. Mr. President, Mr. Chairman, and honored guests, it is my pleasure to present to you Amer Hossain.
Natalie Jones, on behalf of Tarun Kapoor, who prepared these remarks. Many therapeutics work by blocking of proteins' function. However, in many cases, we want a drug-like chemical that can activate proteins, as their loss of function, possibly through genetic mutations, can cause disease. Identifying such drug-like chemicals could lead to new treatments for currently difficult-to-treat ailments, such as Alzheimer's or Lou Gehrig's disease. Natalie Jones, a student in the tri-institutional PhD program in chemical biology, discovered a drug-like chemical that can activate VCP, an essential protein that helps unfold proteins that need to be degraded. Mutations in VCP have been linked to different neurodegenerative diseases, and activating its function could be an effective treatment. Natalie not only designed, synthesized, and tested compounds, but also used cryo-electron microscopy to determine in atomistic detail how these drug-like chemical activators can bind VCP. While inhibitors need to bind a protein in one state to block function, activators must allow the target protein to adopt different conformations needed for function. Natalie therefore characterized multiple different activator-bound states and uncovered a plausible mechanism for how the chemical activator works. Her findings also provide the critical data needed to improve the potency of the drug-like activator. After completing her thesis research, which was supported in part through a Therapeutic Discovery Award she received, Natalie is doing her postdoctoral training at the Cold Spring Harbor Laboratory. While she worked with us, we valued her creativity, dedication, intellect, and passion for chemical and structural biology. We miss her infectious enthusiasm for her work and for soccer, a sport she excels at, and we're particularly impressed by her Bollywood dancing skills. We wish her all the best and look forward to her continued success. Mr. President, Mr. Chairman, and honored guests, it is my pleasure to present to you Natalie Jones.
Congratulations.
Danielle Lynn Keahi started her journey in science at the Kamehameha Summer Science Institute for Cell and Molecular Biology. The program prepares native Hawaiian high school students for laboratory work by teaching them molecular biology techniques. From that point on, every summer, Dani spent doing or teaching science in places including her native Hawaii, Jackson Laboratory in Maine, University of Oregon, and Academy of Athens, Greece, eventually landing as an undergraduate student in Lee Rubin's lab at the Harvard Stem Cell Institute. There, she studied the role of the transcription factor NeuroD6 in the specification of dopaminergic neurons in the midbrain. We were very lucky to recruit Dani for our PhD program, where she joined my laboratory to study BRCA2, a protein that most of you know prevents breast and ovarian cancer. However, Dani wanted to understand how BRCA2 protects neuronal stem cells, specifically the granule cell progenitors of the cerebellum. These cells are highly proliferative stem cells that during early postnatal development become the most numerous cells in the brain, forming the granule layer of cerebellum. Children who carry pathogenic variants or mutations in both alleles of BRCA2 often develop devastating medulloblastomas, which arise from these granule cell progenitors that somehow, without BRCA2, inappropriately return to the cell cycle, giving rise to the medulloblastomas. Dani wanted to understand how BRCA2, a protein that repairs DNA, is specifically necessary for protecting the granule progenitor cells. She combined my lab expertise in studying DNA repair protein with the expertise of Dr. Mary Beth Hatton's laboratory. Mary Beth, who's here with us too, a preeminent developmental biologist specializing in understanding the cerebellum, including the biology of the granule cell progenitors, became Dani's co-mentor. By combining deficiency of BRCA2 and P53, Dani created a mouse model of medulloblastoma and showed that the mutations driving tumor development overlapped with the areas that were rich in guanine nucleotide, which can form complex secondary structures called G quadruplexes. Dani's data indicate that BRCA2 is necessary to prevent the G quadruplexes from being a block to replication and transcription, or is needed to repair the DNA breaks that appear when replication runs into these G quadruplexes. She also found that PIF1 helicase, which can unwind the G quadruplexes during replication, was highly upregulated in tumors. When Dani removed PIF1 from primary medulloblastoma tumor cells, the cells became more genomically unstable, especially after she treated them with chemicals that stabilized those G quadruplexes. These studies highlight that PIF1 helicase is a potential therapeutic target to treat BRCA2 mutated medulloblastomas and identify G quadruplexes as vulnerability in our genomes that necessitate the function of BRCA2 proteins, even in the brain. Both of our laboratories will miss Dani, and we will wish her success as she starts as a senior scientist at Graviton Bioscience. Mr. President, Mr. Chairman, and honored guest, it's my pleasure to present to you Dani Keahi.
Right, it's me again, and last time I have many students, and I think it's because I have a good lab environment. And just yesterday, Amer showed up in my office, and I didn't see him for 6 months. And he said, I said, "Hey, Amer," and he said, "What are you doing?" And he said, "Do you have any deodorant?" So, and I did have deodorant, so it's a. Anyways. Claire Kenny. Claire didn't ask for the other one, by the way. Claire graduated from Johns Hopkins University before joining the tri-institutional MD PhD program, which led her to start her thesis in my lab in 2017. By now, you probably remember that my lab investigates how bacteria employ the CRISPR-Cas9 system to neutralize viral infections. Claire studied the requirements of the Cas9 nuclease to choose its targets on the viral DNA. The initial characterization of Cas9 indicated that this enzyme cleaves DNA sequences that are followed by a guanosine dinucleotide motif, which is also known as NGG. Claire found that Cas9 can also recognize a more complex motif containing an additional adenosine nucleotide, NAGG. Her work revealed a previously unknown feature of the Cas9 nucleus that is important not only to understand how CRISPR works in the context of viral infections, but also to optimize and improve the gene editing technologies that are based on Cas9. Mr. President, Mr. Chairman, and honored guests, it is my pleasure to present to you Claire Kenny.
Jasper Levering would not want anyone to know about this, but I found it out on the internet. At the age of 19, he was featured in the newspaper as one of Denmark's young talents. In that article, in addition to describing his achievements as a high school student, he was asked to give advice to other young people. So he said, according to Google Translator, "Do not settle for being mediocre. There is a whole world out there to strive for." True to those words, Jasper moved on to study at Oxford, being honored every year and graduated first in class. I must believe it is the same philosophy that also led Jasper to us, Rockefeller University. And extremely lucky for me, he chose to join our laboratory to study a protein named CFTR. CFTR is a chloride channel critical to our health. Loss of CFTR function causes cystic fibrosis, a devastating disease still has no cure. On the other hand, excessive activation of the same protein by bacterial pathogens such as cholera leads to secretory diarrhea, which is itself a leading cause of disease in children worldwide. We wanted to understand how CFTR functions, what are the root causes of the diseases, how the existing drugs work, and finally, whether we can discover new or better drug candidates. Working with incredible intensity, focus, rigor, and creativity, Jasper has made major contributions in each of those inquiries. He identified the specific pathway within CFTR through which chloride ions enter and leave the cell. He measured precisely when and how CFTR changes its form to open this chloride passage. He studied the defects of mutations found in patients and showed how drug molecules partially reverse those defects. Also, through a collaboration, Jasper has identified compounds that can tune the activity of CFTR either up or down. Those compounds open the possibility to develop new therapies against cystic fibrosis and secretory diarrhea. Beyond his own research, Jasper is also an intellectually stimulating colleague. To many of us in the lab, he has been a source of knowledge, ideas, and inspiration. Now he's ready for a new journey. We will miss him dearly. At the same time, I'm also looking forward to learn what he achieves next with his talent and dedication to science. Mr. President, Mr. Chairman, and honored guests, it's my pleasure to present to you Jasper Levering.
So one of the truly great and unparalleled joys of academic science that we're here today to celebrate is following the transformative journey of our students over the course of their thesis. And with Rufei Li, I could not be prouder of how far she has traveled on her scientific journey. So Rufei grew up in Wuhan, China, some 7,481 miles away from where we stand today. She received her undergraduate degree at Peking University in molecular neuroscience before arriving at Rockefeller, where she embarked on a completely new scientific trajectory, transitioning from molecular and cellular studies of the nervous system to considering how the complexity of behavior unfolds at the circuit level. In my lab, Rufei was captivated by the beautiful and tightly intertwined duet that male and female Drosophila fruit flies perform as part of their mating ritual, an intimate conversation with no words, only actions. And while much of the field is focused on the male's behavior due to the fact that he performs an extravagant and showy motor display that serves as an overt representation of his internal arousal state, Rufei chose instead to focus on the far more enigmatic behaviors of females to understand how they actually evaluate a male's performance to decide with whom to mate. So female flies are subtle in their gestures, yet ultimately hold the power in these mating rituals, which seems really a perfect metaphor for Rufei herself, whose understated nature belies enormous power and strength. Rufei has never flaunted her achievements like a peacock, but her brilliance shines as bright as his magnificent tail feathers. Through a series of fantastically creative experiments, Rufei made unexpected observations about the interplay between male and female courtship behaviors and female mating decisions that really reframed our thinking about the principles of sexual selection, opening up entirely new vistas along the way, paths that many in the lab and I think in the field will continue to pursue. So in her studies, Rufei has traveled far, not just geographically and scientifically, but most importantly, personally, emergently as an incredibly effective, insightful, and confident scientist. She's an exceptional experimentalist, intuitive with her insights, and always sharing them generously. But what I really admire most about Rufei is that, like all great scientists, her travels are never complete. She relentlessly challenges herself to rise to her full potential, continuously pushing the boundaries of her science, whether modeling a female behavior or developing new methods to explore their underlying neural dynamics. So the next step of her journey is a postdoc with Lisa Giacomo at Stanford University, which I have no doubt she will continue to chart new territories with her scientific discoveries, and we will very much miss her. So Mr. President, Mr. Chairman, and honored guests, it is my great pleasure to present to you Rufei Li.
Congratulations.
Christoph Neumeyer. From his grad school application, Chris was really a jewel. At the bench, he had mastered many a tool, so we happily accepted him into our grad school. Our expectations were high, so we played it quite cool when his first win on campus came from playing pool. Even before he arrived, his publication list was immense. But once he was focused and always intense. It took less than 4 years before his thesis defense, developing a therapeutic based on antisense. Protein targets are hard with features conserved. What hits one, hits others, it's often observed. But at the RNA level, much less is preserved. His gamble paid off, his success is deserved. For Chris, ending a cancer had quite the allure. Not a treatment, but instead something to endure. His drugs killed this cancer, they might be a cure. Long-term, he'll apply it as an entrepreneur. For those who know him, it will come as no shock that first he will pursue a postdoc in immunology at NYU, just right down the block. I hope it is clear Chris is one to admire. He always aims for goals that are higher. His approach to his work can only inspire. Mr. President, Chairman with no qualifier, I give her Ph.D., Christoph Neumeyer.
Masato Ogishi. After brilliant medical studies in Tokyo, he joined our lab 6 years ago. And he quickly turned out to be an extraordinary PhD student. I say extraordinary, not having adopted the charming American taste for laudatory superlatives. Like other Europeans, I'm more fluent in critical superlatives. This tells you how brilliant Masato has been in the lab. His aptitude include incessant activity, clarity of mind, unique ability to multitask, and an understanding, almost instinctive, of the scientific triad: the problem, the hypothesis, and the approach. He's been not only an extraordinary PhD student, but also an extraordinary human being, because despite these aptitudes and skills, he's been loved by everyone, as attested by the fact that although he has many first author papers, he has 2 or 3 times more second author papers. So what did he discover? The problem in the lab is the problem of inter-individual variability in the course of infection. Why would some people develop infectious disease when others infected with the same microbe do well? For 30 years, students in the lab and postdocs would systematically refuse to study tuberculosis because it's the most difficult problem, occurring in about 5 to 10% of infected people. It's easier or less difficult to tackle a problem with an infection that strikes 1 in a million people. I'm not going to get into details, but it's easier. But Masato immediately decided to tackle TB. And I will single out 2 discoveries briefly. First, he discovered human beings genetically deprived of PD-1 or PD-L1, which are known molecules, in particular in the field of tumor immunology. He discovered that these deficiencies underlie a predisposition to tuberculosis, and he deciphered the mechanism at work. And second, he discovered a completely new molecule called LY9, which he showed controls the production of a cytokine which is important to control Mycobacterium tuberculosis. That was an enigma in the field since 1983 when Carl Nathan discovered gamma interferon. And LY9 controls the production of gamma interferon by T cells. So, Mr. President, Mr. Chairman, guests, I'm delighted to introduce Masato Ogishi.
Congratulations. Thank you very much.
Carl Palmquist. Carl grew up in Long Island and came to Rockefeller as a PhD student after attending Swarthmore for his undergraduate studies. Carl was actually the first student to reach out to me and Alan. It was maybe the day after we moved to New York City and even before he had applied to Rockefeller. Carl expressed a precocious interest in undertaking integrative studies that span multiple scales in order to study complex phenomena. Carl's thesis work focused on the mechanisms of self-organization in collectives of cells during follicle patterning in the skin. His initial efforts went towards developing a novel ex vivo, or out-of-the-body, reconstitution of skin pattern formation. Collaborating closely with a research assistant, Carl established a first-of-its-kind platform to reveal the self-organizing waltz of the fibroblasts, the cells in our skin. Utilizing this platform, Carl uncovered an elegant interplay between cells and their extracellular matrix that generates a supracellular fluid, and this spontaneously forms regular patterns. This work really underscored the regulatory power at this supracellular scale during the formation of our organs. In the last 2 years of his PhD, Carl teamed up with Sichen Yang, another of today's graduates, and they investigated the role of signaling molecules called morphogens during follicle formation. Together, Carl and Sitchin discovered a new role for morphogens that only manifests at this supracellular scale, and so couldn't be determined through analysis of molecular or single-cell processes alone. Carl lives for the lab, but does so with a noble modesty. He is a force to be reckoned with, but a discreet one. He's an unassuming rebel. For his impressive thesis work, Carl received what I'll term today a Rockefeller graduate student hat trick. He earned the NSF Graduate Research Fellowship, the David Rockefeller Award, as well as the Weintraub Graduate Student Award. Carl is currently a member of the Institute for Advanced Study, and going forward, Alan and I have every expectation that he'll build on his already impressive contributions, evolving the ideas he developed here through extensions into his current studies of soil ecosystems. Mr. President, Mr. Chairman, and honored guests, it's my pleasure to present to you Carl Palmquist.
It is with great pleasure that I introduce Gabby Panicia. Gabby graduated summa cum laude from Stony Brook in 2019 with honors in biochemistry. During her undergraduate years, she worked with Lori King at Stony Brook to exploit CRISPR-Cas9 technology to target latent murine gamma herpesvirus infection. This experience really solidified Gabby's interest in viruses and basic research. After 2 more summer research internships, Gabby arrived at Rockefeller in 2019 and began her laboratory research rotations. Her arrival in our lab unfortunately coincided with another, SARS-CoV-2 and the COVID-19 pandemic. The university shut down in mid-March 2020, leaving Gabby unable to perform experimental work. She spent her days off-site reading and thinking about possible projects to pursue when she could return to the laboratory. She also used this time to apply for both the National Science Foundation Fellowship and the prestigious Howard Hughes Medical Institute Gilliam Fellowship, which aims to train and enable the next generation of scientific leaders. Gabby received both these fellowships and has taken full advantage of the Gilliam experience to hone her leadership skills. So getting back to the lab, and given some difficulties encountered with an initial project that she had hatched on West Nile virus, Gabby pivoted to work on SARS-CoV-2 as part of a group effort to conduct a genome-wide arrayed CRISPR knockout screen to uncover host factor interactions influence SARS-CoV-2 replication. For many years, the lab has been interested in understanding how cells protect themselves from virus infection, and nature's remarkable solution, the interferon system. From these screens, pro- and antiviral host factors influencing virus infection and interferon responses were identified. Gabby then followed up on one of these, a cellular factor, host phospholipid scramblase, that blocks infection both without and with interferon pretreatment. Through dedication, persistence, and careful experimental execution, she was able to determine that phospholipid scramblase 1 actually exerts its antiviral activity early on when SARS-CoV-2 is attempting to sort of enter a potential cell. On a personal note, Gabby has been a joy to have in the lab. She radiates energy and enthusiasm and loves science and life. She will be moving on to Harvard for her postdoc and continuing in the infectious disease arena. However, this time she will be trying to understand how a fungus that infects flies manages to turn them into zombies whose altered behavior helps transfer the fungal spores to new hosts. This is really just yet another example of amazing biology, and I'm confident that Gabby will succeed brilliantly in this phase of her career, and I look forward to following her future contributions to science. Mr. President, Mr. Chairman, and honored guests, it's my pleasure to present to you Gabby Panicia.
Congratulations.
Josue Regalado. My student, Josue Manuel Regalado, is brilliant, but what has always struck me even more about him is his deep sense of humility and work ethic, and his genuine desire to understand the complexity of brain and behavior. He reads avidly from history to philosophy to science, but he's not complacent about this knowledge. Rather, he's often troubled by what he doesn't know, or what we as a field don't know. Should neuroscientists be studying animals and their behaviors in more natural, ethologically relevant environments? What are mice actually thinking and feeling in our tasks? And do we need newer models and ways of thinking about the brain to fit this behavioral complexity? Thus, Josue was always ahead of his time, not asking the next natural question for a publication, but thinking years ahead about a career, about the arc of his career in science. Josue grew up in Reno, Nevada, attended the University of Nevada for undergraduate training, and had a strong foundation in research as a Barry Goldwater Scholar and HHMI research student at Columbia University prior to joining us as a PhD student at the Rockefeller University. For his thesis work, Josue focused on the question of how animals learn. Even though my lab studies memory, Josue knew that how we learn affects how we remember. He wanted to understand the factors that affect learning, including the role of effort, motivation, and value. By developing new mouse behavioral paradigms and pairing this with high-resolution brain imaging, Josue made a fundamental discovery: that neural signals in a part of the frontal cortex of our brain scale with the motivational state of the animal. That is, he found that as animals escalate their desire for a goal or a reward, the activity in their frontal cortex continues to ramp until that goal is achieved. This is a striking neural correlate for a complex brain state and has important implications for how motivational signals are routed to downstream areas of the brain to dictate learning, memory, and decision-making. In addition to his main thesis project, Josue was a co-lead author on another entirely separate project in the lab, as well as a key contributor to almost every ongoing project in the lab, a remarkable testament to his scientific drive and collaborative spirit. Beyond the lab, many of us know what an invaluable asset he's been to Rockefeller community through his efforts as co-president of the Rockefeller Inclusive Science Initiative, RISI. For many years during his PhD, Josue put an enormous amount of thought and effort into leading community efforts, working collaboratively with university leaders as we constantly strive toward a more inclusive and just campus. Looking back, it's incredible to me how much he managed to get done for science and society. Much like his Mexican immigrant parents, Josue has a boldness to seek new opportunities, take risks along the road less traveled, and build a collective success. I can't wait to see all that he will accomplish in his journey ahead. Mr. President, Mr. Chairman, and honored guests, it's my pleasure to present to you Josue Regalado.
Matthew Reynolds. Cells in our body physically engage their surroundings through the cytoskeleton, a network of proteins that allows them to generate and detect forces. Defects in the cytoskeleton produce aberrant cell shape and cell movement, which are associated with developmental disorders and metastatic cancer. As the cytoskeleton's functions are inherently mechanical, forces play a central role in regulating cytoskeletal proteins and the dynamic networks they build. We currently lack a fundamental understanding of how forces alter these proteins, making it unclear exactly what has gone wrong when the cytoskeleton malfunctions in disease, a prerequisite for developing therapies. Forces are difficult to precisely control at the molecular scale, and visualizing how they alter proteins is therefore an ongoing challenge. Matt Reynolds is a computational biophysicist who embraced the messiness of this problem. He developed new machine learning-based approaches for detecting rare molecular signatures in electron microscopy images. With these tools, he was able to hone in on subsets of molecules experiencing similar forces, enabling him to analyze their structures. Working with experimentalist colleagues, he provided a first glimpse at how bending forces alter polymers composed of the protein actin, the core building blocks of the cytoskeleton. He used similar approaches to produce the first atomistic structures of protein bridges known as crosslinkers, which link multiple actin polymers together to construct higher order networks. This work resulted in several high-impact publications for which Matt was recognized with the American Society of Cell Biology's Alan Hall Award in 2022. Matt is currently pursuing a short postdoc in the lab while he figures out his next move. He will be missed for his extraordinarily keen intuition and skill in extracting meaning from noisy data, which have elevated nearly every project in the lab. Beyond his encyclopedic knowledge of image processing, he also maintains a personal ranking of every pizzeria he's ever visited in the city, both of which he is always delighted to share. Mr. President, Mr. Chairman, and honored guests, it is my pleasure to present to you Matt Reynolds.
Adam Rosenzweig, presented on behalf of Professor Sean Brady. Adam is part of the Tri-Institutional Chemical Biology program. When he joined the lab, we were just finishing the development of a new method for accessing antibiotics from nature. Adam has applied that novel discovery approach to the identification of next-generation antibiotics that we hope can one day be used to head off the growing antibiotic resistance crisis. Most of the antibiotics in use today were discovered more than 50 years ago by studying the metabolites produced by bacteria that were easily cultured from a diversity of environments around the world. The approach that Adam used during his graduate studies didn't rely on bacteria produce those antibiotics that they do in nature, because they're actually not very good at doing that in the lab. He instead used DNA sequencing and bioinformatics to predict new antibiotics encoded by bacterial genomes and then total chemical synthesis to create these never-before-seen antibiotics. During his time in the lab, Adam mastered this complex process and successfully applied the approach to the identification of multiple new antibiotics that seem thus far to avoid the development of resistance, which is the key issue threatening the current arsenal of antibiotics in clinical use. Although it's still early, I believe that this work will lead to the development of a new generation of critically needed improved antibiotics. It's been a pleasure to have Adam in the lab, and he will be missed by his lab mates. At different times during his graduate career, he served as our lab's social chair, organizing regular lab lunches, and the lab benefited from his extraordinary skills as a baker. I wish Adam the very best in his future efforts to apply what he has learned in graduate school to the private sector. Congratulations, Adam, and I hope you will continue to nurture your dual interests in science and the culinary arts. Mr. President, Mr. Chairman, honored guests, it is my pleasure to present to you Adam Rosenzweig.
Anna Reba came to Rockefeller via Williams College in the post-baccalaureate studies at the NIH with a clear vision for her PhD to explore the neural mechanisms that generate behavioral variation, ultimately focusing on how genetic variation shapes brain wiring, a mechanism that lies at the very core of what defines us as individuals, each with our own unique perception of the world. Identifying heritable differences in brain circuitry across national populations is not an easy task. To gain traction into this fundamental problem, Anna focused on how male Drosophila detect and perceive female pheromones, a process that is central to the recognition of an appropriate mate. Anna discovered that beyond the well-described differences in pheromone sensitivity that are apparent across different Drosophila species that serve to reinforce reproductive barriers between them, there's also striking variation even within different natural strains of a single species in how males use female pheromones to dynamically guide their mate choice. Anna astutely recognized that she could leverage this rich natural variation as a powerful inroad to shed light on the sites of heritable plasticity within the nervous system. And using an array of really clever experimental techniques, Anna showed that quantitative variation in inhibition at just a single neural locus predictably determines whether males will promiscuously chase all females or selectively hone in on just an appropriate mate. So through this beautiful comparative analysis, Anna revealed how males of different strains tune their responses to the same chemical cues and shed light on the basic neural substrates of behavioral variation that serves as a foundation for individuality and evolutionary selection. So Anna is herself an exceptional representative of her species. Working with her has been really a true joy because of her, not only of her, beyond her brilliance and humility, she has a distinct scientific voice that is creative and eloquent. Yet what I really admire about Anna is just how generous she is in sharing her voice and passion for science, both within the lab and beyond. So for several years, Anna has co-directed the Summer Neuroscience Program, which serves under-resourced New York City high school students by providing them with a 2-week immersive research experience on campus. And this really helped Anna to recognize that there are many ways to impact and transform the landscape of scientific discovery. And there's no doubt that Anna has really been at the forefront in her desire to transform science at multiple levels, both through her commitment to outreach and her rigorous and creative research. I'm so proud to have been witness to Anna's own evolution as a scientist, emerging as one of great clarity and insight, and look forward to seeing her imprint on the scientific landscape in the years to come. So, Mr. President, Mr. Chairman, and honored guests, it is my great pleasure to present to you Anna Riba.
Congratulations.
Hi, Arjen Skeepers. Arjen joined our lab immediately when we arrived at Rockefeller in 2016, first as a visiting master's student from University of Utrecht, and then as a PhD student here in the program. His 8 years in our lab have been stellar by any measure and have earned him prestigious awards like the Boehringer Ingelheim Fund's scholarship and the Brinsteel Award for PhD research in the life sciences. As I'm sure you all know from experience with the SARS-CoV-2 vaccine, several shots are needed for one to achieve maximal protection. This is because the level of protective antibodies in blood increases dramatically from one shot to the next in a phenomenon we know as immune memory. Arjen's PhD work has made profound contributions to our understanding of this whole process. His 3 published studies were instrumental in revealing the intricacies of the response to booster shots at the level of the B cells, the cells that produce the antibodies that go into the blood. His work shifted our understanding of the nature of secondary B cell responses and revealed the cellular mechanisms behind a phenomenon known as original antigenic sin, which can limit our ability to respond to variant boosters encoding different strains of the virus. His contributions has been as wide as they have been deep. As a postdoc in the Rudensky lab at Memorial Sloan Kettering, I am certain that Arjen will go on to make a similar mark on the T-cell field. Mr. President, Mr. Chairman, honored guests, it's my pleasure to present you Arjen Skeepers.
Yanis Tazi, I'm sure, is the person in this room with the largest number of academic degrees, and that is before he even receives the one today. Born in Morocco, Yanis studied at 2 Paris universities, in Montreal and Singapore at Columbia University, NYU, excelling everywhere while obtaining advanced degrees in mathematics, computer science, data science, computational biology, and business. For his graduate work, Yanis applied himself and his tremendous computational skills to the better and deeper understanding of facial expressions. Charles Darwin famously suggested that our facial expressions and those of related animals serve to express our emotions. They can be elicited by facial expressions of others through social contagion, and thus serve as an important means of communication in social species like ours and in species lacking speech. Yanis has made many innovative contributions to the understanding of both the perception and the generation of emotional expressions. First, he used advanced computer vision approaches to characterize facial movements and find an embedding for different facial expressions. Second, he built artificial neural network models that process both expression and identity, beating state-of-the-art approaches in the process, and then analyzed these networks to understand how they do what they do. All of this was done with an interest in understanding how the brain accomplishes these tasks, which led him, in the final part of his thesis work, to analyze data obtained from the networks of the brain controlling facial movements to identify a new coding strategy we had not known before. Yanis did all of this at lightning speed. So if you want to think of him as a hero in your Marvel universe, he's the Flash, but somehow behind a computer. So Yanis is a polymath and a world citizen and will now go out and bring his many talents into the worlds of academia and industry. So that's in the presence of his family who came all the way here from Morocco to witness this. I am happy and proud to proclaim, Mr. President, Mr. Chairman, and honored guests, it's my pleasure to present to you Yanis Tazi.
Congratulations.
Rebecca Timpson. Unfortunately, Kivanch Bursoy had to be out of town. He sends his regrets, but I was honored to know Rebecca both in the classroom and through a thesis committee, so it's a pleasure to read Kivanch's remarks. Rebecca Timpson arrived at Rockefeller with a deep passion for mechanistic biology. Though trained as a biochemist, her fearless nature led to explore diverse areas, from mouse genetics to cancer biology. Her thesis addressed a fundamental question: why do mammalian cells have antioxidants? Focusing on the most abundant antioxidant, glutathione, Rebecca delved deeper into its role in mitochondria, the cell's powerhouses that also generate damaging free radicals, as Rockefeller professor Albert Claude had previously discovered. Her research, alongside colleagues, unveiled how glutathione enters mitochondria to exert its profound protective influence. But Rebecca's brilliance extended beyond this. Inspired by bacterial species, she designed a unique tool to precisely control glutathione levels within cells and organisms. This approach revealed a surprising truth. Too much of this beneficial antioxidant can be detrimental to health. Beyond her scientific talents, Rebecca is known as one of the most helpful scientists I've ever encountered. Her willingness to lend a helping hand without hesitation or self-interest has been invaluable to the many people around her. It's bittersweet to see her graduate, but we'll miss her in the lab, and we're excited for the many discoveries that await her. Mr. President, Mr. Chairman, and honored guests, it's my privilege to present to you Rebecca Timpson.
Cesar Vargas. I believe that great scientists are often people who have made accomplishments in another walk of life. Cesar is one of those. He started out as a gymnast and won multiple dozens of awards in gymnastics in his early childhood and teenage years. It's a discipline that requires lots of discipline, patience, and effort and creativity, which Cesar brought to science. He did his undergraduate training at Vanderbilt University, majoring in neuroscience and the history of art. He worked with Vivian Casagrande on changes in brain connectivity in autism model of mice. Then he joined us here at Rockefeller the year I arrived back here myself to start a new lab here. He was the second graduate student to join my lab since arriving here at Rockefeller. He took on the ambitious project to characterize vocal communication circuits and function in mice for traits that are once thought to be unique to humans, this ability to produce imitated sounds. And we found that mice had some rudimentary principled functions in their brain that indicated that there was something there that could be a little bit human, but Cesar wanted to investigate that. He investigated the direct connection from the cortex to the neurons that control the vocal organs and found that there actually were sparse connections that were functional. The cortical areas controlling the voice in mice thought to be unique to humans. It just wasn't all direct as it is in humans. It was a partial direct connection and mostly indirect. He also found that the motor cortex is more mixed in its functions than the traditional homunculus model that many of you learn about, the hand, one location, the foot in the other. Instead, Cesar found that they're even sometimes the same neuron connected to neurons that control the voice, that control the jaw, and control the hand, all at the same time. So his study is revising the homuncular organization of the brain. So this all contributed and is consistent with what we call the continuum hypothesis of vocal learning. It's not the haves or the have-nots. There's some gray areas of in between, and mice are somewhere in between. To do all of this, Cesar also had to be creative with not only molecular tools, but software. He worked on developing a software that analyzes mice vocalizations in real time, doing analysis not only offline, but as the mice sings, and then can track that information and decide what to do to give the mice some food for interesting vocalization or not. So these studies have implications for using mice as rudimentary models of certain aspects of spoken language in humans. He's published several papers from this work, and several are under review now. He's now, actually, before I get to where he's going, outside of science, Cesar has been a person who has really been in tune with the culture of science. Being a person of Mexican heritage, he was one of the first persons here to start, or the founding members of RISI, Rockefeller Inclusive Science Initiative, that were concerned about issues of underrepresentation in science. He also was instrumental in helping form new policies that surrounded the civil unrest and what we were going to do about it here following George Floyd's death here at Rockefeller. Well, not here at Rockefeller, but the policies. All right. And so I'm going to give a big shout out to Cesar and his partner in crime here, Sway, to say that I really think they helped change a lot of the culture here at Rockefeller. So Cesar's leaving is bittersweet. He will be sorely missed. He's doing a postdoc at Sam Sober's lab at the University Emory, this time taking what he learned in mice and translating it to songbirds. So I'd like to thank his thesis committee, Jim Hudspeth, Priya Rajase Thupathi, and Yutaka Yoshida, our external expert. Mr. President, Mr. Chairman, and honored guests, it's my pleasure to introduce Cesar Vargas.
Congratulations. So proud of you.
We're coming into the home stretch. Hang in there. It is my happy and sad privilege to read Professor A. James Hudspeth's graduation statement for Camilla Villasante. I have had the pleasure and honor of collaborating with Jim and Camilla and Xinyu Dong, a younger graduate student. Unfortunately, Professor Hudspeth was recently diagnosed with glioblastoma, had major brain surgery, and is now undergoing radiation. I wish him a prompt, steady, full recovery. Here is his statement. Hearing is enormously important in our lives. When sound waves push hair cells of the inner ear, the protein protocadherin 15 in the filamentous tip links of hair cells stretches, and opens mechanically sensitive ion channels. The excited cell signals the brain. That's how you hear. In her doctoral research, Dr. Villasante determined the mechanical properties of individual dimers of protocadherin 15 with an optical trap. Her ingenious experiments showed not only that protocadherin 15 can control the channels, but also that a mutation in protocadherin 15 makes the protein less stable and causes hearing loss. Camilla's work provides valuable insight into how hundreds of tip-link mutations interfere with human hearing. In addition to her scientific and clinical activities, Ms. Villasante has advocated for women and minorities in medicine as a board member of the Female Association of Clinicians, Educators, and Scientists, and the Association of Diverse Physician Scientists in Training. She also led diversity initiatives and mentored underrepresented students. Her high intelligence, demonstrated research ability, and contributions to advocacy and education make Ms. Villasante an excellent prospect for a successful career in biomedical research and clinical practice. Now I speak for myself. Camilla impressed me with her quick understanding, willingness to put serious effort into trying something new, patience, and care in communicating her results to someone vastly ignorant like me. During our joint work, just incidentally, Camilla also got married. Congratulations. I share Jim's assessment of Camilla's happy prospects for a successful career in whatever she may choose. I also look forward to helping Shinue. Mr. Chairman, Mr. President, honored guests, it is my great pleasure to present you Dr. Camilla Villasante.
Samuel Williams. Sam is a rising 8th-year MD PhD student in the Tri-Institutional MSTP program. He's a Native American from the Lumbee tribe of North Carolina, and Sam is the first self-identifying Native American in the 50-year history of this Tri-Institutional program. And so for me and his family, it's indeed a proud moment. During his time in my lab, Sam produced a great thesis centered on a hugely debilitating skin disease termed hidradenitis suppurativa. In this work, Sam discovered a fundamentally new way that anaerobic bacteria can stimulate epithelial cells to become direct inflammatory mediators or players and orchestrators of cellular immune responses that create autoimmunity. Sam was a great colleague to have in the laboratory, and he will be highly missed. We all expect him to become a leading physician scientist as he completes the Tri-Institutional Biomedical Program. Mr. President, Mr. Chairman, and honored guests, it's my great pleasure to present to you Samuel Williams. Congratulations.
Sichen Yang. It is my pleasure to present Sichen Yang for his doctoral degree from the Rockefeller University. Sichen grew up in China and came to Rockefeller as a PhD student after completing an undergraduate degree at Tsinghua University in China. Sichen was the lab's first graduate student. His thesis project focused on the role that signaling molecules play in generating tissue structure. His work challenges accepted dogmas as to how such molecules influence morphogenesis, and provides an original conceptual pathway for addressing the ancient question of how the body is formed. Our laboratory was founded on the realization that self-organization amidst collectives of cells can generate the morphological patterns seen across the body. Rather than being puppeteered by genetic or molecular control mechanisms, the formation of our tissues may be the result of epigenetic processes that occur beyond the scale of single cells. However, if cellular self-organization can create structure, what roles do essential genes, such as morphogen signaling molecules, play in such processes? This is the question that Sichen boldly addressed in his graduate work. He found that morphogens such as BMP and FGF allow collectives of cells to adopt distinct biophysical properties at supracellular scales. Through biophysical measurements, he showed that these properties were emergent, since they could not be readily measured at single-cell scales. Furthermore, he showed that these supracellular phases can mechanically interact to power morphological events, such as follicle budding in the skin. More broadly, his work indicates that in systems where cell collectives self-organize, it may not be possible to understand the function of molecular signals by the methods of genetics and biochemistry alone. Sichen's work has shaped the way our laboratory approaches morphogenesis, providing conceptual and experimental pathways for approaching collective cell behavior, while not losing sight of the richness that occurs at subcellular scales. Sichen has been offered postdoctoral positions in several leading labs in the areas of cancer and immunology, where he plans to bring his understanding of the emergent properties of collective cell behavior to bear. Our lab has benefited deeply from Sichen's joyful approach to science and life. We have always appreciated his interest in all Rockefeller has to offer, ranging from classical music to pickleball tournaments. He lives a whole life, which no doubt has been a big factor in helping us see morphogenesis more holistically. Mr. President, Mr. Chairman, and honored guests, it is my pleasure to present to you Sichen Yang.
We have no Z's. So, last but not least, Yihao Yang. At Rockefeller University, I've had the pleasure to mentor truly special young scientists through their graduate education. In many different ways, each has shined as rising star. Yihao grew up in China. He is a first-generation immigrant, first college graduate, and first Ph.D. in his family. Channeled by his curiosity of science in high school, Yihao came to the United States in 2013, where he began working in Catherine Plath's laboratory at UCLA to investigate how the X chromosome status is established. Upon graduating from UCLA in 2016, he transitioned to Rockefeller to enter our Ph.D. program here in 2017. In his application to Rockefeller, Yihao expressed many particular interests, none of which involved my laboratory. Hence, I was fortunate when he asked for a rotation in my group and shortly thereafter joined my laboratory for his graduate studies. In the early days, Yihao worked closely with a postdoc, Nick Gomez, who was one of the top experts in bioinformatics in my laboratory at the time. Yihao soon took over this position. In a collaborative effort spearheaded initially by Nick and then by Yihao thereafter, they focused on a fascinating question that has roots in the development of the mammalian embryo, namely, how are tissues specified when they all have the same DNA? And how are they able to continue to perform their dedicated tasks thereafter? My laboratory had shown that right after cells decide that they're going to make skin rather than brain, they face the decision, if they're skin, whether or not to continue to make the epidermis that covers our body surface or instead make hair follicles. At the heart of this decision is a transcription factor called SOX9. Yihao discovered that SOX9 is an amazing protein that on the one hand can bind and open up chromatin in the hair follicle stem cell genes, and on the other it pulls away the key chromatin remodeling and transcription factors that had been the epidermal stem cell genes. Fortunately, we have a balance in this process, so we don't just make hair, nor do we just make epidermis. However, Yihao learned that when SOX9 becomes unleashed and the levels get too high, a cascade of events begin to happen that lead to skin cancers. This landmark study was published in a major scientific journal, and it serves as a paradigm for how other tissues of our body faithfully do their chosen tasks and don't get coaxed into making the wrong decisions. His findings have broad implications for other tissues and for future cancer therapeutics. Yihao's ability to work comfortably with high-throughput technologies and focus on big-picture questions landed him a prestigious position at Altos Pharmaceuticals in San Diego. On top of it all, he continues his interest in Japanese, in bodybuilding, and in photography. Yihao, I look forward to what comes next in the future. I wish you the very best for what is going to be, no doubt, an exciting future. Mr. President, Mr. Chairman, and honored guests, it's my pleasure to present to you Yihao Yang.
Congratulations. Well done. Very proud of you. Everyone seems to be here. Graduates, please rise. By the authority of the Regents of the New England Commission on Higher Education, which they have vested in the trustees of the Rockefeller University and by them is delegated to me, I confer upon you the degree of Doctor of Philosophy, which admits you to all the rights and privileges thereunto pertaining. Congratulations. You may be seated. Now we turn our attention to the 2024 recipients of the Doctor of Science honoris causa, Francis H. Arnold, Freeman A. Hrabowski III, and Michael Mann. These three individuals have shaped the scientific landscape in myriad ways through their vision, innovation, and leadership. It is a tremendous privilege to recognize them here today. I'll begin by recognizing Frances Hamilton Arnold. She's the Linus Pauling Professor of Chemical Engineering, Bioengineering and Biochemistry, and Director of the Donna and Benjamin M. Rosen Bioengineering Center at the California Institute of Technology. At age 11, Frances Arnold wanted to be a heart surgeon, But that plan evaporated when she found she couldn't tolerate the sight of blood. She was a highly curious and rebellious young individual, and at age 15, she was skipping school in Pittsburgh to hitchhike to anti-war protests in Washington, D.C. Her parents found this hard to take and issued an ultimatum that she stop, upon which she moved out, rented an apartment, and worked odd jobs to pay the rent. Her continued truancy earned her poor grades in general, but her teachers wondered, how is it that she's earning straight A's in math? They were sufficiently curious that at one point they asked her to solve these problems on the chalkboard to prove that her test results were legit. They were. She went on to ace the SAT exams and wrote a college application essay about life on her own that was likely unique to Ivy League admissions officers. She applied to the Princeton Mechanical and Aerospace Engineering program and has said that she applied there because she thought it was the easiest program to get into, particularly since there had literally been no females in the program, and she was accepted. The program suited her as it had very few requirements and enabled her to take a broad array of courses in addition to math and physics. During her time as an undergrad, she took a year off to live in Spain and Italy. Her father, a physicist, helped her get internships in Madrid and Milan, doing stress tests analysis for nuclear reactor pressure vessels. Boredom with this task left her eager to find out what other things mechanical engineers could do, and she returned to coursework with new zeal. The oil crisis of the 1970s was broiling, and renewable energy caught her attention. She thought that she could help solve one of the biggest problems of the day. So after graduating in 1979, she spent a year at the Solar Energy Research Institute in Colorado. But she didn't find the work sufficiently interesting. So she applied and was accepted into the Chemical Engineering Graduate Program at the University of California, Berkeley, intending to develop biofuels. At matriculation, someone who was clearly paying attention noticed that she had never taken a class in chemistry, which might be an impediment to the development of her career. As a consequence, she spent her first year taking all of the undergraduate courses in chemistry, at which she excelled. By this time, however, Ronald Reagan had been elected, and funding for alternative energy research had dissipated. But another opportunity caught her attention. The revolution using recombinant DNA was starting to transform biology, and the possibilities seemed truly endless. She obtained her Ph.D. working with Harvey Blanch, defining and applying principles of affinity chromatography to the purification of proteins, while learning biochemistry, enzymology, and molecular biology and evolution. At graduation, she realized a few things. She didn't want to have a boss. She wasn't particularly interested in financial profit. So in 1987, she joined Caltech and was promoted to the faculty, and she's been at that institution ever since. At the time, the discovery of new enzymatic capabilities in nature for a wide range of industrial applications were being sought by a number of approaches. Extreme environments in nature, all the extremes of temperature, pH, salinity, the presence of heavy metals, etc., had proved to be good places to find microbes that had evolved their enzymes to be active in extreme environments. Bioengineering approaches were being pursued to try to develop proteins with novel enzymatic properties. Many would-be protein engineers attempted so-called rational design, attempting to go from a protein's atomic level structure determined by X-ray crystallography to conceive specific changes in the protein sequence that might produce desired changes in activity. This method, however, mostly failed. Our understanding of determinants of protein folding and catalysis were woefully underdeveloped. Francis approached the problem from a fresh angle. Rather than trying to learn and implement these elusive rules of protein folding and catalysis, she recognized that over eons, evolution had done perfectly well at producing these enzymes simply by natural selection that were well adapted to the niches of the organisms these activities were found in. She intuited that evolution could also work under novel selective conditions that had never been encountered in nature and could be used to evolve tolerance to novel environments as well as to evolve novel chemical activity, enzymatic activities. And while evolution in nature can be extremely slow, she thought it was obvious that one could greatly accelerate the process by using the emerging DNA technologies to introduce random mutations into proteins and use sensitive assays to identify which of these engineered mutations had effects that headed the protein in the desired direction. Selecting these mutants for further iterations of mutation and assay, one might be able to predictably and efficiently produce enzymes that are optimized for the desired activity. This directed evolution procedure does not rely on any understanding of the protein's function, nor does it forecast which amino acid swaps will be successful. Suffice to say, the idea was met with wide skepticism from many standard bearers of protein engineering. But guess what? The strategy worked beautifully. In 1993, she published a paper that showed how sequential random mutagenesis of a protein-cleaving enzyme that normally works great in aqueous environments inside the cell, but has almost no activity in organic solvents, could acquire activity in this environment by random mutagenesis and sequential selection for improved activity. After several rounds of selection, she evolved a protease that was 250-fold more active in this organic solvent, dimethylformamide, a novel chemical environment. A beautiful aspect of the result was that the amino acid perturbations that conferred the desired properties would not have been predicted to exert their beneficial effects. Over the last 30 years, this method of directed evolution of proteins has been widely used for diverse purposes. Some have found application in industry, like enzymes for use in cold water laundry detergents, or in medicine, in glucose sensing for glucose monitors in diabetes, and in pharmaceutical and textile manufacturing, disease diagnostics, and more. In 2005, Frances launched her own company, still in business today, that is working toward making making large quantities of sustainable aviation fuel. Even more remarkably, she has cultivated enzymes that do chemistry that may not have been found in nature. For example, in 2016, Frances's lab used directed evolution to generate the first enzymes that create carbon-silicon bonds, a pairing of elements that is not seen in nature. Silicon-containing products permeate our lives, and their manufacture by conventional synthesis requires platinum, which is mined through toxic practices. Biocatalysis, which is now possible thanks to Frances's breakthrough, provides an environmentally clean alternative. Manufactured materials containing silicon can on their own contaminate the environment. Compounds called volatile methyl siloxanes, for example, have proven so valuable and versatile that their production has reached megatons annually, but their accumulation and ability to escape into air pose safety concerns. Just this January, Frances's group reported that it had developed enzymes that break man-made carbon-silicon bonds in this particular class of compounds, suggesting a path to making these substances biodegradable. Committed to deploying science to serve people and the planet, Frances accepted an invitation from President Biden to co-chair the President's Council of Advisors on Science and Technology, or PCAST. Under her leadership, PCAST has reported on patient safety, wildland firefighting, extreme weather, greenhouse gases, semiconductors, and more. The importance and impact of Frances's work has been well recognized. She received the 2018 Nobel Prize in Chemistry, the National Medal of Technology and Innovation, the Millennium Technology Prize, and the Charles Stark Draper Prize for Engineering. She's also the first woman to be elected membership in the trio as a member of the U.S. National Academy of Engineering, the U.S. National Academy of Sciences, and the U.S. National Academy of Medicine. From the above, it's clear that Dr. Arnold is thinking a lot about sustainability. She has said as much, and I quote, Chemical engineers must be responsible for greening of chemical processes. We have to be responsible for making better foods, providing clean water, tackling climate change. In the past, chemical engineers mainly worked in the fossil fuel industries or for chemical companies that converted fossil fuels into chemicals and materials. But now the profession is broadened dramatically, and we're leading the transition to clean energies, clean food, clean agriculture, and clean chemistry. We make things. We make all these things, and we're responsible for doing it better. Francis Arnold, your pioneering development of directed evolution of enzymes awakened scientists in academia and industry to a world of new possibilities in biological catalysis and chemical biology. Your continuing work has led to environmentally sustainable manufacturing methods for pharmaceuticals, novel biofuels, and more. It is Rockefeller's great honor to confer upon you the degree of Doctor of Science honoris causa. Francis, please rise to receive your hood and diploma.
Oh gosh, that was so long. I better keep this short. My mother couldn't have done better. Thank you. This is for the grads, and I'm sorry to be standing with my back to you, but I'm speaking to you and to the families, because it was my little brother, Eddie, who introduced me to biochemistry when I was struggling to find my career path. I'm always struggling at something. And this was in the 1970s. And thanks to him, I got hooked on this amazing field of biology and the chemistry of the biological world. And so I ended up going to UC Berkeley at the beginning of the DNA revolution. And all this new technology of recombinant DNA was coming out. Such a glorious experience being at Berkeley. And you know, the science wasn't too bad either. But the point is that protein engineering was not a thing when I was studying. As you heard, I had a degree in mechanical and aerospace engineering in the middle, when our country was in the grip of an energy crisis. So I said, okay, nuclear power would be interesting. And then, of course, the 3 Mile Island disaster put a powerful brake on its further development. So I jumped into solar energy until the election of Ronald Reagan kind of changed the job prospects in that field. So, you know, moving around a bit, I had 30 years of evolutionary exploration, trying different things, under my belt before I became an assistant professor. No boss, you know, I'm in charge. and set out with my goal to make protein engineering an engineering discipline. This was a time when no one, including myself, knew how to do it. At least to make something useful. But I could see that proteins could help solve so many problems if we could only engineer them to serve our needs, and just as evolution has tailored them to serve biology's needs. So I used this magnificent design process called evolution. Now at Rockefeller, you're at the forefront of advances in the biomedical sciences. Thanks to researchers like you, we can cure genetic disease, we can give dying person new organs, we can battle the scourge of cancer with ever more powerful therapeutics. And these are the fantastic achievements of modern medicine and biotechnology. The problem is they are available to too few. They don't materially help the planet, nor do they move the needle on the dramatic decrease in life expectancy that we've seen in this country for the last few years, driven by violence, drug addiction, pollution, poor nutrition, and poor public health. These are the killers, but sadly they get little funding and little attention. And I implore you, grads, to use your talents and your science to lay these scientific foundations that would someday solve these problems. And far too little attention is paid to the planet killers of climate change and pollution, or to devising sustainable biological solutions to fuels and chemicals and materials and agriculture, to figuring out how we thrive alongside our co-inhabitants, the animals, the birds, the insects, the greenery, the oceans, and so much more that we just take for granted. I've spent a significant fraction of the last 3.5 years pondering how science and technology can serve American citizens as co-chair of PCAST. Here I was thrown into a whole new world of policy for science, but more importantly, of science for policy. How does science make good policy? And I hope that you grads will remember that science will not thrive without the support of your fellow citizens, and that your science has no audience, purpose, or meaning without them. So I'd like to share a couple of thoughts from when I took on this job in January 2021. My belief is that our highest responsibility in each generation is to preserve our fragile planet, prepare for the future, and pass on a better world. Science-based decision-making has always been our most powerful tool for meeting that responsibility, and perhaps never more than today. As climate change, pandemics, loss of biodiversity, as these loom, we look to science and technology to save this precious jewel of our planet so that we might pass it on to future generations intact and in good health as it was passed to us. But science is not just the cold solving of problems. It's a warm and beautiful exploration of the unknown, an expression of human curiosity that propels us forward and allows us to fulfill our most important responsibilities. And the moment we fail to nurture it, we resign ourselves to living in the past, and we lose the chance to guide the future. But when we put science back to the benefit, to working for the benefit of people, we're making a society that is worth passing on to our children and to our grandchildren. So graduates of this jewel of an institution, where you have been given the gift of science, with science we can make possible what seems impossible. So please make science for people and for our planet. Thank you.
Thank you, Francis, for those inspirational words. And now we're delighted to honor Freeman Alphonsa Hrabowski III, President Emeritus of University of Maryland, Baltimore County. When Freeman Hrabowski's teachers assigned math problems, he usually asked for more. That request was not always welcomed by his classmates. But his parents knew that they could make it easier to get him to go to church if they would bring along math to chew on. One day when he was 12 years old, he was sitting in a pew puzzling over some algebra, and he heard that day's speaker, Martin Luther King, who said that an upcoming children's march right in his hometown would help them get better schools. This got Dr. Hrabowski's attention. He knew that the books in his classes and the schools he attended were not like those that white kids had. He persuaded his parents to let him participate in the 1963 Birmingham, Alabama Children's Crusade. Although the demonstrators endured horrific treatment during the protest and afterward in jail, the experience empowered him. It summoned an activist spirit and imbued a powerful dose of Dr. King's message. The world tomorrow could be better than today, but only if Freeman helped make it so. He went on to major in mathematics at Hampton Institute, graduating at age 19, and then continued to the University of Illinois, where he earned a master's degree in mathematics and a Ph.D. in higher education administration and statistics. He aimed to employ statistics to understand science, technology, engineering, and math, STEM programs in higher education, hoping to propel more people of color into science. Among the barriers was skepticism that Black people should even be there, a phenomenon that he had encountered firsthand. In an advanced math class, the professor entered the classroom, looked at Freeman, the one Black student among white men, and said pointedly, "This is set theory," implying that Freeman did not belong in the class. This continued, and after a few such episodes, Freeman preempted the instructor when he walked into the room and said pointedly to him, "This is set theory, right?" That didn't happen again. After earning his Ph.D., Dr. Hrabowski began an academic career and quickly rose into leading administrative positions. He joined the University of Maryland, Baltimore County as vice provost in 1987. UMBC caught his interest in part because it had been the first campus in the state to accept students of all races. He had visionary ideas and the talent to implement them. He was named president of UMBC in 1992. Throughout his career, he has championed the belief that meeting humankind's challenges will best be accomplished by incorporating wide-ranging perspectives. He has enriched the scientific enterprise by cultivating aptitude in individuals from varied backgrounds, including those from marginalized groups. Toward this end, he has enlisted people who have traditionally held power, yet seek to do the right thing. This venture cultivates the best talent across racial and cultural boundaries to strengthen problem-solving capabilities. It also boosts public trust in science and medicine by bringing role models into every community. When Dr. Hrabowski took the helm of UMBC, a predominantly white and Asian institution, conventional wisdom dictated that only historically Black colleges and universities were appropriate for educating Black people. But UMBC, under his guidance, showed that people of any race can help people of other races to excel. With appropriate guidance and resources, Black students shine not just as occasional geniuses, but in large numbers. Furthermore, the university has transferred its strategies for training and retaining science undergraduates from underrepresented groups to individuals in all disciplines, of all backgrounds. More than a third of the Black and Hispanic students at UMBC are first-generation college students. UMBC crafted a blueprint that now provides a model for the entire country. The university's approach grew from its experience with a program that Freedmen and philanthropists Robert and Jane Meyerhoff launched in the late 1980s. The Meyerhoff Scholars Program aimed and aims to increase diversity among future leaders in science, technology, engineering, and math by supporting individuals who intend to pursue a Ph.D. or both an M.D. and Ph.D. degree. It originally included only Black men, but has since expanded. Freeman and his colleagues assembled numerous synergistic elements to foster success. High expectations rule at UMBC for the students and for the professors. Faculty members have developed strategies to rethink the instructional processes. For instance, they consider the possibility that low test scores in a class reflect poor teaching effectiveness that needs to be rectified. A strong apprenticeship component fuels individuals' academic journeys. Lab heads recruit and nurture the next generation. Crucially, Freeman built an academic community in which each individual can flourish. Students collaborate to achieve academic excellence, studying together and sustaining one another emotionally and professionally. They stay in contact through graduate school and beyond. These ongoing connections counteract forces that tend to drive underrepresented minorities out of STEM career paths. True to Freeman's original vision, he and the faculty have ensured that UMBC harnesses data to assess what's working, what's not working, and how to improve. The results have been astounding. Under Dr. Hrabowski's stewardship, UMBC has become a powerhouse in educating STEM students who not only make it through college, but become some of the best in their fields. Most remarkably, the university has generated more Black bachelor's degree recipients who have gone on to earn Ph.D. degrees in the natural sciences or engineering or MD PhDs than any other institution in the nation. And when you stop to think about the institutions it's competing with in that endeavor, this is truly remarkable. Some of these individuals now serve on the faculties of the best U.S. research universities. The success of Dr. Hrabowski's passion and approach has sparked the development of programs well beyond their boundaries. For example, inspired by his achievements, the Howard Hughes Medical Institute, one of the world's largest and I dare say the best scientific philanthropies, introduced the Freeman Hrabowski Scholars Program in 2022. This $1.5 billion initiative aims to advance diversity in science by supporting outstanding early career faculty who are committed to inclusive lab leadership. Awardees prioritize excellence in their own research while they create positive lab environments, promote diversity and equity through mentoring, and serve as models within and beyond their home establishments. In 2011, the Washington Post and Harvard Kennedy School's Center for Public Leadership named Dr. Hrabowski one of the 7 great American leaders. And the following year, Time named him one of the world's 100 most influential people. President Obama recruited him to chair the President's Advisory Commission on Educational Excellence for African Americans. The American Council on Education awarded him a Lifetime Achievement Award and followed up by naming him the organization's inaugural Centennial Fellow. He's a member of the U.S. National Academy of Engineering, and in 2023, the National Academy of Sciences awarded him the Public Welfare Medal for his extraordinary use of science for the public good, making him a member of the National Academy of Sciences. Freeman Hrabowski, we recognize you today for your brilliant, fearless, and transformative leadership of UMBC. During your 30-year tenure as president, you built a remarkable university community that offers equality of opportunity for all, emphasizing mentorship, collaboration, and wise investment of resources to achieve an outsized impact. Your moral compass and success as a leader of higher education is an inspiration to colleges, universities, and organizations across our nation and to all of us on the Rockefeller campus. It is a great privilege for Rockefeller University to confer upon you the degree Doctor of Science honoris causa. Freeman, please rise to receive your hooded diploma. Over here. Congratulations. Let me, here, I'll give you this. I've got to take a photo. Congratulations.
Thank you very much, Richard. Thank you. When I was in graduate school at Illinois, one of my professors was from Poland, and he came in and he said, he was calling the roll. It was a very small class, and he said, Hrabowski, and I raised my hand, and he said, no, Hrabowski. And I said, this is America. I am Hrabowski, all right? I'm delighted to be here, Mr. President, Mr. Chair, and faculty, and others. I want to start by doing something that's not appropriate. You're never supposed to turn your back on the audience, but I want to look, as Frances did, I want to look into the faces of the graduates for just a minute. I just wanted to look into your faces because, as she said, I'm going to be talking to you, to your families, but really to you, all right? So you've got to imagine I'm looking at you. So, got it? Got it? Okay? All right. Today, first of all, thank you. I am deeply honored to be receiving this degree. And I accept it on behalf of my colleagues from the university, from UMBC, and students. And one of my wonderful vice presidential colleagues is here somewhere, Lynn Schaefer. I'm just delighted she decided to come up. And it speaks to, her coming speaks to the importance of relationships. And today, for me, this is a celebration not only of scientific achievements, but of the human experience. I start with the notion that First of all, if you didn't know it, families, you're going to have to take a test on all the science before you get out of here today. But I watched your faces, and while most of you probably are not in science, so you're not like my friends Leslie and Eric and Mary Beth, who knew the science and were really cheering and got it all, most of you were pleasantly dignified, having no idea, other than maybe when they mentioned the mosquitoes and a little pain and a few things. Am I right? Right? But you were so proud of your family members who are part of this distinguished group at the university that known for producing so many Nobel laureates. This institution represents the best in the world. And students, graduates, if you don't know it, only about 1% of human beings in the world have earned a Ph.D. And even in our country, it's only been about 2%. And the areas that are least accessible for most people will be in the sciences. We know that it's very difficult for people to go, not just from high school, but to college and college on to the next level. And so I had several thoughts. Number one, it was 61 years ago that I sat in jail in Birmingham as a nerdy little math major. I love math. How many of you love math? Let me see that here. Now let me turn around. How many of you love math? Let me see. Pretty nerdy group like my campus. Thank you very much. Most Americans will ask, how can you put love and math in the same sentence? You know that, right? But for me, math has always been at the heart of learning. I see in nature math. I see it in everything that we do. But somehow I was in jail because I did want a better education. And I remember Dr. King saying, I was 12, he said that the arc of the moral universe bends slowly towards justice. But the other part of his message that has been so important to me that I give to you, graduates, that arc only bends towards justice if we make it do so, if we are empowered to make it happen. It's what we do as individuals, as institutions, that can make a difference. The question I have asked for 60 years, so how do I help more people in general, and then more black kids, more women, more others, to love math and science and to excel, the Latinos? And so I was so honored today to see the students from Mexican-American backgrounds and Latinos and the first Native American. Big deal, huge deal. Huge deal. Because what happens is when people hear about that, others say, it can happen. And children know it can happen. Some of you know that I'm most proud of our students and Kismikia Corbett, a little kid from North Carolina, from rural North Carolina, who co-led the team at NIH to produce the Moderna vaccine, became the first black woman in the world to create a vaccine. And so she's now in the faculty at Harvard. But when she walks into a room and little girls of any race see her, and they say she created a vaccine, what does it say? I can do it too. Kafri Zarasa Mary Beth, who is from Duke and who just recently published a paper on the African-American brain, on a big study being done at Hopkins, who's now full professor there in neuroscience. Again, the notion that we in the human race must understand that to have the kind of trust that we want to see from the public must see people from all backgrounds, first generation college, people of all races, people from all over the world. Because the challenge we face right now, in addition to the climate issues that Francis mentioned, and I'm so honored to be here with Francis and Michael, is that beyond the global issues like that, it's how many people don't trust science or medicine. I spoke at a university in the last year and a half, and somebody was going to mention that one of my students had helped to produce this Moderna vaccine, and they decided to take it out because if they had mentioned the vaccine, 40% of the families would have protested. This is what we see in some parts of our country, if you get the challenge that we face. And so I'm going to challenge you, graduates, to know that you are among the best in the world and the most privileged, because you've not just earned a Ph.D., but in the biomedical sciences, that leading place in the world. And there's a saying that you've heard, of those to whom much is given, much is required. And I challenge you to think about how you connect to the larger society, to pull more people into this work. And there are 2 or 3 thoughts. One, be inspired by your own story. It's not just about you. It's about all the people, the relationships you have. You're looking at your families now. You know you would not have made it without the faculty, your mentors, family, and friends. And it will always be that way. Number 2, and most important, never lose the curiosity. Hrabowski, the Nobel laureate in physics, said that when he was growing up in New York, all of his friends' mothers would ask the kids at the end of a school day, "What'd you learn in school today?" He said, "But not my Jewish mother. My mother would say, 'Izzy, did you ask a good question today?'" And that practice of encouraging the curiosity made the difference. And then never forget the questions. Francis and I were talking about it's asking questions about the world, but about ourselves, to keep learning about ourselves. It's so important. I'm still asking those questions and trying to learn more and more. I decided to study French in my late 60s, several years ago. And my students laughed and they said, "Don't you think you're kind of old, right?" And that was all the energy I needed, right? So, mes étudiants et moi, nous parlons le français chaque jour. Mon professeur est originaire de Normandie. Il est normand, il est très rigoureux, right? And it's Apollinaire who says, "Le joie venait toujours après la peine." La joie venait toujours après la peine. Meaning what? That the joy comes after the struggle. When I finished Illinois in 75, that's been 50 years, students, the question on my mind was, will I be okay? And all of you right now, as accomplished as you are asking yourselves, will I be okay? And of all the things I can tell you, you're gonna be okay. You're gonna be okay. Just keep learning. Samuel Beckett said in one of his books about the dancing of bees, that the character said, "Here's something I could study all my life and never fully understand." Because the more bees you see the bees dancing, the more they're communicating with each other. And he began to understand it, but yet he realized the more he learned, the more there was to know. And it's the idea you keep learning. You never stop learning. And there's my challenge to you, to remember the bees, to remember the questions, and then remember the essence of this institution about science and humanity, of all the things I can say. Aristotle, another great scientist, that's a joke, folks, you can laugh, said, "Excellence is never an accident. It is the result of high intention, sincere effort, and intelligent execution. It represents the wisest option among many alternatives." And then he said this as I close, something that has always given me goosebumps. He said, "Choice, not chance, determines destiny." And there is the question, what will you choose? Thank you all very much. Thank you.
Thank you for those remarkably inspiring words. And for our final honoree today, I am incredibly pleased and proud to introduce Michael Evan Mann, Presidential Distinguished Professor in the Department of Earth and Environmental Science at the University of Pennsylvania and Director of the Penn Center for Science, Sustainability, and the Media. Now, in grade school, Michael tells me that he knew the timeout room very well. He was a wise guy, always challenging authority. He also got into playground fights. Someone would call him Shorty or pick on one of his friends, and Mike would take on offenders twice his size and whatever number was there as needed. When the principal called his mom, called her in, wanted to talk about him getting into fights all the time, she pushed back. She recognized his passion to stand up for what was right, and she didn't want to squelch that instinct. Eventually, Mike would come face to face with one of the biggest bullies on the planet. And of course, I'm talking about the fossil fuel industry. And he did not back down. Born and raised in Amherst, Massachusetts, where his father was a math professor, in junior high school, Mike found Carl Sagan's Cosmos. He learned about the ancient Greek scientist Eratosthenes, who 2,300 years ago discerned that the Earth was round, and he accurately calculated its circumference using only the angles of shadows cast at the same time in two cities on the same meridian. These and other stories captivated him, fueled his natural wonder about the universe, and gave Mike the sense that scientists are explorers, and that was something that appealed to him. Math and science came easily, and by high school he was flourishing. On Friday nights, he'd head to the computer room where he ate pizza and programmed late into the night. He was accepted at University of California, Berkeley, where he double majored in physics and applied math, and then continued to Yale for doctoral work, intending to work on theoretical condensed matter physics. Although the problems in that area were stimulating, they didn't grab him. Their applications failed to touch the big picture world problems that Mike yearned to tackle. While thumbing through the program catalog, hoping to find a topic that sparked his interest, he came across Barry Saltzman, a luminary in the field of weather and climate. With Saltzman, Mike could focus on his math and physics skills on the Earth's climate system. He decided to study the impact of natural phenomena on climate variability and switched to Saltzman's group for his Ph.D. Upon completing his thesis, he moved to the University of Massachusetts in his hometown and joined Ray Bradley as a postdoctoral fellow. There he investigated the effects of, for instance, volcanic haze and solar output on Earth's surface temperatures. Thermometer logs went back only about 150 years, so to document the fluctuations that occurred before then, he exploited indirect measures whose properties reflected meteorological features. Tree ring growth, for instance, chemical evidence in corals, ice cores, and ocean sediments. Dr. Mann and his colleagues developed robust statistical methods combining multiple proxies to reconstruct the historical climate record year by year and region by region around the planet. A dramatic moment came when they assessed surface temperature for the Northern Hemisphere over the previous 600 years. They found that the 20th century had been the warmest in the previous 600 years, and they reported this in a paper in Nature in 1998. Furthermore, 1990, 1995, and 1997, all within the most recent decade, held the highest individual year temperature records. The team soon added another 400 years to the analysis, and the sharp rise grew more striking as the flat portion of the graph reached back for an entire millennium. These data sent an unmistakable message. The planet was heating up, and the simmering had started around 1900. Furthermore, that timing was closely correlated with a prodigious increase in the release of carbon dioxide into the atmosphere, most of which was the consequence of the Industrial Revolution and the dramatic increase in the burning of fossil fuels. Although Mike's initial interest centered on natural causes of climate variability, his findings about the temperature surge forced him into the realm of anthropogenic human-caused influences. The topic of climate change had been gaining prominence and contentiousness as well in public discourse, and his high-profile publication put Mike under a spotlight. The graph's hockey stick shape, with a long arm and then an upturned blade, became an iconic depiction of global warming. Mike was interviewed on multiple news networks, and he told the New York Times, quote, "Our conclusion that the warming of the past few decades appears to be closely tied to emission of greenhouse gases by humans and not by any of the natural factors." This was rather modestly stated, I have to say, but it nonetheless promoted and spurred major reactions. As the first author and most vulnerable member of the team, at that time he didn't yet have a faculty position, much less tenure, Mike became a target for climate change deniers. After he was chosen as a lead author of the 2001 Intergovernmental Panel on Climate Change report, attacks ramped up. That influential document featured the hockey stick graph in its summary for policymakers and highlighted the dramatic influence that human activity, especially fossil fuel burning, was exerting on the planet. Conservative media pundits assailed him, and Republican lawmakers called him on the carpet to testify before congressional committees. They demanded extensive documentation that dated back to the beginning of his scientific career. This harassment soaked up tremendous amounts of time and energy. Mike had to solicit legal advice rather than focus on research, teaching, and other professional activities. And despite the impediments, he persisted and showed that the lower temperatures stretched back not just 1,000 years, but 2,000. A dozen independent reconstructions of the Northern Hemisphere temperatures by others validated his results, concluding that the Earth was warming and the rate of increase was accelerating. Over the last 20 years, Mike's conclusions from the 1990s has become increasingly obvious. Limiting analysis to the last 174 years for which reliable thermometer measures are now available, the National Oceanographic and Atmospheric Association reports that the 10 warmest years in this period have all occurred in the last 10 years, with 2023 being the hottest. In fact, each of the last 47 years has been warmer than the annual average of the 20th century. Everyone who has been paying even modest attention to the last several years has seen the impact of climate change on the evening news or in your own lives. More extreme weather events are occurring, and the unprecedented, for example, scale of wildfires in the western U.S. and across the northern Canadian forests, increased frequency of severe tornadoes, as we see on the morning news every day in the last month, and marked warming of the oceans and the melting of sea ice are all spawning increased number and intensity of hurricanes. In late 2009, hackers purloined emails written by Mike and other climate scientists. By broadcasting snippets of these emails out of context, participants in the climate change denialism movement accelerated their efforts to discredit Mike and his colleagues, claiming that despite massive evidence to the contrary, that his research was fraudulent. Eventually, the risks became physical. Emails and anonymous internet postings threatened his life and those of his family members. He received a letter including a mysterious white powder. Undeterred by the turmoil, Mike has continued studying Earth's climate system, including human-caused change, and the results document that our greenhouse gas emissions are shifting rainfall patterns, shrinking glaciers and sea ice, and inciting extreme weather events. In addition to his scientific works, Mike has demonstrated an unwavering commitment to and talent for explaining climate science and controversies. In addition to op-eds and commentaries, he has written 6 books for the general public, including one for children entitled The Tantrum That Saved the World. He co-founded the website realclimate.org, which aims to educate the public and journalists, and he has persevered in his efforts to protect scientists from political attacks due to their work. In February of this year, after a lengthy court case, he won a landmark lawsuit against 2 bloggers who continued to accuse him of scientific fraud without any evidence. The court ultimately ruled in Mike's favor, concluding that the defendants, quote, made false statements with maliciousness, spite, ill will, vengeance, or deliberate intent to harm, and awarded him substantial punitive compensation. Despite all of these challenges, he remains passionate, encouraging, and optimistic, noting that change is possible. He points out that over the last 50 years, the U.S. enacted legislation that markedly cleaned up our waterways and air quality and dramatically reduced acid rain that was devastating eastern forests and lakes. Moreover, the international community has taken collective action that has mitigated the hole in the atmospheric ozone level that science showed was caused by chlorinated fluorocarbons and other ozone-depleting substances. He notes that throwing up our hands in defeat only guarantees the accelerated destruction of a livable planet. The lengthy list of Mike's honors and awards includes the Tyler Prize for Environmental Achievement, dubbed the Nobel Prize for Environmental Preservation, and the Leo Szilard Award of the American Physical Society. In 2023, the American Humanist Association named him Humanist of the Year. He's an elected member of the U.S. National Academy of Sciences, and just this year he was elected a foreign member of the UK Royal Society. Michael Mann, today we recognize your transformative contributions to climate science and your cogent efforts to educate the public about climate change. Your pioneering application of the tools and principles of math and physics to analyze historic data collected from multiple sources confirmed the impact of 20th century human activity on our climate on a hemispheric and global scale. We also celebrate your measured responses to climate controversy as a speaker and author. Rockefeller University is immensely honored to confer upon you the degree of Doctor of Science honoris causa. Mike.
Well, thanks so much, President Lifton. Thanks to the Faculty Honorary Committee for this great honor. Congratulations to the graduates. I can't see you, but I can feel you with my spidey sense. I'll get back to that later. And let me also say, it truly is an honor to share the stage here today with both Francis and Freeman, who, to quote my good friend Bill Nye, are working to change the world. And that's what we're really talking about here. And you'll hear some of the same themes that both of them spoke to in my comments here today. At a time when science is so critical to the great challenges we face as a civilization, it's especially meaningful to receive an honorary degree from an institution like Rockefeller, that has made singular contributions to the furthering of societally relevant science. Rockefeller University has, of course, shown particular leadership in the area of health and medical science. And while I'm not a life scientist myself, some of my best friends are. Includes my wife, Lorraine Santee, who's actually a cell biologist, a card-carrying cell biologist, and my good friend Peter Hotez. Some of you may know that name. In fact, Peter and Tony Fauci, when you heard those stories from President Lifton about some of the things that I went through more than a decade ago, they unfortunately sort of portended similar events that we're seeing today with scientists studying COVID-19 and vaccines, and I'll come back to that. As I mentioned, my good friend Peter Hotez, who's a notable graduate of this illustrious institution. In fact, I was joking to Peter that we're both now Rockefeller University graduates. Peter's a friend and a colleague, and we're currently writing a book together due out in mid-2025, the title of which is Science Under Siege. And make no mistake, science is under siege today, as is fact-based discourse writ large, and truth, as I've stated before, has never been more fragile, nor has our civilization or our planet. It's up to all of us to defend each of them before it truly does become too late. There's a war being waged against science by politicians and polluters and petrostates and posers and pretenders and propagandists. You can see I like alliteration, and bad actors in our press. Social media and now AI have been weaponized to undermine faith in governmental and civic institutions, our traditional media, expert opinion, and yes, of course, scientists. That includes public health experts like my friends Peter Hotez and Tony Fauci, who've worked tirelessly to inform the public about the science of COVID-19, only to find themselves the targets of a vicious, politically motivated anti-science movement. And if you're reading the headlines, this is literally playing out today. Demands for emails being taken out of context to try to discredit Tony Fauci. And And of course, it includes climate scientists. I found myself in the center, as you've already heard, of this fractious debate over climate change the better part of 3 decades ago when my co-authors and I published the original hockey stick curve. It visually communicated the threat of our ongoing addiction to fossil fuels, and I found myself targeted by industry front groups and their enablers who sought to discredit me and my work. I've remained resolute in defending my science and more generally the science of climate change from these bad faith attacks. And I have indeed embraced the opportunity that that has given me to inform what is arguably, the greatest challenge that we face, the public discourse over the greatest threat that we face today. Those who fail to learn from history, it is said, are doomed to repeat it. So let me speak to a dark episode from our past, highlighted in the recent Oscar-winning film Oppenheimer. I speak of the Red Scare of the late 1940s and early 1950s, when American scientists associated with the Manhattan Project were investigated for suspected communist ties or leanings. J. Robert Oppenheimer himself, as most of us know, lost his security clearance with devastating personal and professional consequences. Another scientist who plays a far more brief but still memorable role in the film, and by the way, you heard I received the Leo Szilard Award of the American Physical Society a few years ago. Leo Szilard plays an important, brief but very memorable role in the film as well. But I'm going to talk about a different physicist, Edward Condon. Condon was the director of the National Bureau of Standards and president of the American Physical Society. He testified in 1948 before the HUAC chair, J. Parnell Thomas. Thomas variously referred to Condon as Dr. Condom, the weakest link, and even the missing link. The inquisitor at a later related hearing chastised Condon. It says here that you've been at the forefront of a revolutionary movement in physics called quantum mechanics. It strikes this hearing that if you could be at the forefront of one revolutionary movement, you could be at the forefront of another. President Harry S. Truman defended Condon at the opening address of the 1948 annual meeting of the American Association for the Advancement of Science, where he spoke at his side. Continuous research by our best scientists, Truman warned, may be made impossible by the creation of an atmosphere in which no man feels safe against the public airing of unfounded rumors, gossip, and vilification. That was 1948. Truman's words couldn't resonate more today, 3/4 of a century later. The fact that anti-science has been embraced and weaponized so fully by one of our two major parties, frankly, is grave cause for concern. As Peter and I argue in our forthcoming book, these attacks themselves represent an existential threat, for they undermine our ability to address the twin great challenges of our time, the climate crisis and the growing threat of deadly pandemics. So when we find ourselves, we find ourselves in a battle of Tolkien-esque proportions, facing an unprecedented assault on science, reason, and fact-based discourse, the stakes simply couldn't be greater. Scientists, academics, and institutions of higher learning don't only have a right to speak up and speak out, they have a duty to do so. And so I leave you with the words of the great philosopher Stan Lee. With great power comes great responsibility. And I speak now, and I'm using my spidey sense so I can see you behind me, even though you might not realize it. I speak to this year's graduates. I'll actually turn around. You have great power. You have great power, and you have an opportunity to use it. As my good friend Bill Nye likes to say, change the world. I like to tweak that a little bit. You have the power to change the world for the better. So please do it. Thank you.
What an amazing trio of moving and important talks. Thank you so much. And now, I'd like all three of you to rise. You're good. You're good. Francis Arnold, Freeman Hrabowski, and Michael Mann, by the authority of the Regents of the New England Commission of Higher Education, who they have invested in the trustees of the Rockefeller University and is by them delegated to me, I confer upon you the degree of Doctor of Science honoris causa, which admits you to all the rights and privileges thereunto pertaining. Congratulations. Yeah.
Can we give a standing ovation to the class?
Okay. Well, honorary degree recipients, Chairman Bill Ford, graduates, faculty, staff, students, honored guests. This concludes the 66th Convocation of the Rockefeller University. So on behalf of the graduates and honorands, I'd like to invite all of you to the ensuing reception. I gather there's been rain outside. Is that true? Yeah. Okay. Well, so instead of having this reception be outdoors, it's going to be just across the walkway in the tall glass building on the opposite side of the marble walkway as you depart the building. This is called the CRC, and there are people right in the lobby who will help you get there. So now I'd like to ask all of our guests to remain at your seats until as we exit the building and to please stay until the marchers have exited the auditorium. So once outside the auditorium, the graduates and honorary degree recipients, along with everyone else who's with me on the stage and the faculty presenters, will assemble for a class photograph in the Abbey Reception Hall in front of the Women in Science portrait, which I think is very fitting for today's event. So following the photograph, the graduates will join their families and friends at the reception. And now I'd like to ask everyone to please stand. I trust we have music that is going to start momentarily.
Keep it running.